Hi and welcome back to freesciencelessons.co.uk. By the end of this video, you should be able to describe how to carry out chemical tests for carbohydrates, proteins and lipids. And remember that lipids is another word for fats. Now, this is a required practical and it contains a lot of detail. It could come up in your exam, so it's really important that you learn this. I should point out that all of the chemicals in these tests are potentially hazardous, so safety goggles must be worn throughout. Okay, so first we take our food sample and we grind this with some distilled water using a mortar and pestle. We want to make a paste. We then transfer the paste to a beaker and we add more distilled water. We stir this so the chemicals in the food dissolve in the water. Next, we filter the solution to remove suspended food particles. At this stage, we can now test our solution for the chemicals present. We're going to start by testing for carbohydrates. This includes starch and also sugars such as glucose. To test for starch, we place 2 centimeters cubed of our food solution into a test tube, and we add a few drops of iodine solution, which is an orange colour. If starch is present, then the iodine will turn blue-black. However, if there's no starch, then the iodine will stay orange. Now, testing for sugar such as glucose is a bit more tricky. Again, we start with 2 centimeters cubed of our food solution. We then add 10 drops of Benedict solution, which is a blue colour. We place the test tube containing our solutions into a beaker, and we half fill the beaker with hot water from a kettle. We now leave this for around 5 minutes. If sugars are present, then the Benedict solution will change colour. The colour of the Benedict solution gives us a very rough idea of the amount of sugar present, but it cannot tell us the exact amount. A green colour tells us that there's a small amount of sugar. A yellow colour tells us that there's more sugar present, and a brick red colour tells us that there's a lot of sugar present. Now, the Benedict's test only works for certain sugars such as glucose. Scientists call these reducing sugars. The Benedict's test will not work for sugars which are non-reducing such as sucrose. Okay, let's look now at how to test for protein. We take 2 centimeters cubed of our food solution, and we add 2 centimeters cubed of Biuret solution, which is a blue colour. If our solution contains protein, then the Biuret solution will change colour from blue to a purple or pink colour. The final test we're going to look at now is for lipids. Now, in this test, just like before, we grind our food with distilled water using a mortar and pestle. However, unlike the other tests, we do not filter the solution when testing for lipids. We transfer 2 centimeters cubed of our